Welcome to this explainer. Today, we're skipping all the fluff and diving straight into the hidden errors that are literally costing you top band grades in Cambridge Biology 9700. If you've been banging your head against a wall trying to secure those elusive final marks in topic 2.2, saturated and unsaturated fatty acids, well, you are in the exact right place. We're going to break down the biological architecture of this topic, look at exactly what the examiners are hunting for, and show you how to structure your answers so scientifically that you'll never lose an easy mark again. So think about a typical fatty acid exam scenario. Take a mental guess right now. What is the single tiny error that consistently destroys scores before the examiner even finishes reading your first sentence? Is it a botched calculation? Maybe a gap in complex scientific reasoning? Most students think it's a lack of broad knowledge, but the actual root cause? It's way more specific than that. Just listen to this direct quote pulled straight from the principal examiner reports. Answers on 2.2 are often too vague. Candidates consistently confuse key terminology. Boom, that establishes our high stakes right out of the gate. Terminology confusion is the absolute fastest way to bleed easy marks. The Cambridge assessment framework prioritizes analytical depth over superficial breadth, meaning if your terms are even a little bit vague, your entire explanation completely falls apart in the eyes of the examiner.
All right, Section 1, Core Concepts and Definitions, Securing the Foundations. Let's fix that critical terminology gap right now before you even attempt to evaluate complex data.
Here's the deal. Examiners demand absolute precision. You can't just gesture vaguely at things like single bonds or mutter something about animal fats. You must clearly state that saturated fatty acids have hydrocarbon chains with only single carbon-carbon bonds, and that they hold the maximum possible number of hydrogen atoms. Always define your terms precisely before you try to justify a complex causal relationship. It proves to the examiner that you actually possess the core biological knowledge required. And this brings us to the comparative analysis requirement, which is huge. You have to state both similarities and differences using specific comparative connectives to unlock those higher tier marks. For instance, don't just jump in and say, unsaturated fats have double bonds. No, establish the similarity first. Both have a carboxyl group attached to a hydrocarbon tail. Then you hit them with the difference. Unsaturated chains contain one or more carbon-carbon double bonds, which creates kinks in the chain. And it's exactly those kinks that make them fluid at room temperature. You want the secret to literally jumping a grade boundary here? It's cross-topic connections. The principal examiner reports explicitly note that integrating topic 2.2 with other areas of the syllabus, like explaining how those kinks in unsaturated fatty acids actually increase cell membrane fluidity, is the hallmark of a significantly higher scoring candidate. It shows you're treating biology like an integrated system, not just an isolated checklist of facts. Moving on to Section 2, the Statistical Analysis Framework. Let's talk about selecting the right test. You've absolutely got to commit this table to memory. Correctly choosing between a t-test, chi-squared, or correlation test is a totally non-negotiable step in your procedural framework. Look, if the exam asks you to compare the means of two distinct groups of fatty acids, you use a t-test. Dealing with categorical data, like the simple presence or absence of a trait, that's chi-squared. And if you're trying to figure out how one variable affects another, you apply a correlation test. Take a breath and lock those three distinct tests into your brain right now. In just a second, I'm going to reveal the exact examiner clue that flips a 0-mark answer into a top band score. But first, we need to look at the actual framework that makes that clue super obvious, because without a structured approach, even a glaringly obvious hint will slip right past you when exam panic sets in. Which brings us to Section 3. Deconstruct, solve, and evaluate. This is your top band framework. High-scoring candidates don't guess. They follow this sequence every single time. 1. You start with the core biological concept. 2. You inject your data from the stimulus material. 3. You layer in your scientific reasoning. And 4. You finish up with precise examiner wording. You identify what's given, break it into logical steps, solve it systematically, and check your logic. Let's actually see this in action. Consider this severely flawed student answer. Fatty acids affect health. Yikes. Notice how it completely lacks specific context and entirely forgets crucial experimental units. It's totally superficial. It doesn't specify if we're talking about a saturated straight chain or an unsaturated kinked chain. It doesn't quantify the effect. There are 0 units, and it completely ignores whatever specific context was provided in the source material. An answer like this, 0 marks. Now contrast that by strictly applying our framework. Saturated fatty acid concentration in moles per decimeter cubed shows a positive correlation. Look at how this improved top-end answer systematically secures every single method mark available. It names the specific biological concept, includes the exact units, uses correct statistical terminology with positive correlation, and demonstrates evidence-based reasoning. That is exactly what the academic benchmarks for this syllabus are demanding.
All right, Section 4, Top Examiner Warnings. Let's run through the top 3 traps examiners are practically begging you to avoid, pulled directly from the marking trenches.
First, never be vague. Second, obsessively check your units. And third, ensure every single point is locked firmly into the specific context of the stimulus material you were given. Look, if the question gives you a graph about sunflower oil at different temperatures, your answer must specifically reference sunflower oil and those exact temperatures. Do not just regurgitate generic textbook facts about unsaturated fats being liquid at room temperature. You have to use their data.
And keeping with that theme, remember this. It only takes exactly one major out-of-context error to completely derail your critical analysis and lose those high-level evaluation marks. Always prove your claims using the exact experiment provided on the page.
Let's wrap things up with Section 5, Final Exam Strategy Challenge. It is time for a rapid-fire viewer challenge to see if you can accurately apply the statistical methodology we just covered.
Pause for a second and predict this. If an exam question asks you to evaluate complex data using our framework, what is the very first thing you must identify? You have the biological concept and you have the data right in front of you. But what actually triggers which statistical test you're going to use and dictates the direction of your scientific reasoning?
Here is the diagnostic cliffhanger. You could apply the formula flawlessly, use the perfect units, pull the exact context, and still lose massive marks if you misinterpret the hidden cause. What is it? The command words. If the prompt says describe, you just state what the graph shows about the lipid concentration. But if it says explain, you have to give the biological reasons why those single carbon-carbon bonds are causing that specific effect. If you explain when you were supposed to describe, all that beautiful scientific reasoning is completely wasted.
So, how do you decode those command words and secure that top mark band? Well, make sure to subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCS to excel in your Cambridge exams. And go check out our next explainer right now. But before you click away, ask yourself, the next time you see a lipid structure on your exam paper, are you just going to see a simple drawing? Or are you going to see the exact statistical path to full marks? Let's find out next time.